Assalamu alaikum. So now that we talked about um, fatty acids, let's get into complex lipids now. These can be classified into different types. Okay, so we have storage lipids, meaning that these are used for energy purposes. And there's only one type, triacylglycerols. So basically, whenever we have excess fat in our bodies, they are stored primarily in adipose tissues in the form of triacylglycerols. Now, we also have other types of lipids that primarily they are localized or they make up membranes. And these can be classified into two types. We have phospholipids and glycolipids. Now, these phospholipids, they are of two types. We have glycerophospholipids and we have sphingolipids. Okay. Now, Sphingolipids, there's only one type, and it's called Sphingomyelin. Okay. Now, glycerolipids, there are different types depending on the group that is attached to the phosphate group that is part of the backbone known as glycerol. Now, sphingolipids can be of another type, by the way, which is glycolipids. So basically, so let's review this. So we have storage lipid now. Triacylglycerols, they have a backbone known as a glycerol. Remember glyceraldehyde from sugars? So you have glycerol, and attached to it you have three fatty acids. Then we have phospholipids. Okay, we have two types. We have glycerophospholipids because their backbone is glycerol. They have two fatty acids, a phosphate, that's why they're called phospholipids, and whatever group is attached to the phosphate. And then we have sphingolipids, which is also a phospholipid. So their backbone is not glycerol, its backbone is known as sphingosine. Okay, and there's a fatty acid attached to it, a phosphate, that's why it's called phospholipid, and choline. Now, there's only one type, as I said, and it's known as sphingomyelin. Now, sphingolipids, or you have another type that has a backbone of a sphingosine, which is another sphingolipid, except that it doesn't have a phosphate like in here. It has a sugar attached to it, okay, as well as a fatty acid. So let's talk about triacylglycerols. Okay, the old name, by the way, is triglycerides, but they're known as triacylglycerols now. So they have a glycerol backbone, as you can see in here. So it's the same as glyceraldehyde, as I said before, and with three hydroxyl groups. Now, to each one of them, we can have attachment of a fatty acid. Okay. So if there's only one fatty acid, it's known as a monoglyceride or monoacylglycerol. If you have two fatty acids, it's known as diacylglycerol. Three fatty acids, triacylglycerols. Now the thing is, you can have three of the same fatty acids attached or different. Okay, so you can have combinations. Okay, so if you have the same fatty acid attached to the glycerol, then it's known as a simple triacylglycerol. Otherwise, it would be called a mixed triacylglycerol. Now, note that not only that you have different lengths of the fatty acid chain, but you can have different degrees of saturation. So you can have fully saturated fatty acid, or you can have mono unsaturated and or polyunsaturated fatty acid attached to the same glycerol. Now, and remember we talked about cis and trans double bonds, and we said that if you look back into the structure of the cis double bond, it creates a kink, طاجة. Okay, so it creates a kink, and this kink sort of like it requires it makes the fatty acid needing a space, a large space. And this is actually the idea behind fats versus oils. See, if you have if you have a triacylglycerol with 3 saturated fatty acids, this is a highly compact molecule. Okay, so it's quite hydrophobic. Okay, that's why it is solid. On the other hand, if there is a cis double bond, it looks something like this. Now these are the in red, the oxygens. Okay, so you have a hydrocarbon chain with a cis double bond right here. It creates a kink again, and it creates a lot of space around it. Okay, that's why it is oil. Okay. Now, the idea of triacylglycerols is that we can use them for the production of soap and sabun, and this is the concept behind, or this is the process of known as saponification, the formation of soap. So the idea is that we can take samne or fat in a solid form, and we can using a hydroxyl an alkaline environment, we can separate the fatty acids from the triacylglycerol, creating fatty acids that can be in a conjugated base form that can be associated with a possibly charged sodium ions, for example, forming soap. Now this is the process that also occurs in our body, except that it is enzymatically catalyzed by enzymes known as lipases. So we have specific lipases that separate the R groups, which are the hydrocarbon chains, separating the fatty acids from the glycerol moiety or backbone. So this is how soap works. So what you have in here is that you have two sides of the molecule. You have the hydrophilic head of these fatty acids, okay, which can be exposed to water, and you have the hydrophobic chains, the hydrocarbon chains of the fatty acids. Okay, now these try to hide from water. So what happens is that when you wash your hands, you have the formation of what is known as a micelle. Now that's the idea. That's the best way by which you can actually clean your hands from grease or dirt. Grease, yani duhun. So the best way is to put soap on your hands without water for now. Let the fatty acid chains get mingled and mixed with the dirt, because they are dirt and grease are hydrophobic. So the fatty acid chains of soap would be mixed with the grease or dirt, and then when you apply water to your hands, what happens is that these fatty acids would form a micelle. It looks like a ball, whereby the hydrophilic head, the carboxyl group, are exposed to water, and the fatty acid chains are hiding from water. They try to hide from water. They are hydrophobic, so they trap dirt and grease inside these balls, and that's the best way of washing your hands. So there is a process called hydrogenation. This process is basically it entails or involves, or it results in the removal of double bonds. So what happens is that under certain conditions, these can be removed. These double bonds can be removed, converting an unsaturated fatty acid into a saturated fatty acid. Okay, so now the thing is, this process has been used a lot in industry, and the idea is that they want to convert saturated fatty acids, unsaturated fatty acids to partially saturated fatty acids. Okay, the process is basically to convert polyunsaturated fatty acids to monounsaturated fatty acids, so they can convert these oils into semi-solid products. Okay, except that the problem is that during this process, there is conversion of the cis double bond into a trans double bond. Okay, so if you have complete hydrogenation, you would have fully saturated fatty acids. But what happens if you have partial hydrogenation or as a byproduct, يعني ناتج ثانوي, as a byproduct, you would have the formation of a trans double bond. The problem with trans double bonds is that our bodies cannot handle these trans double bonds. Okay, so they tend to accumulate in our blood vessels, and that what causes risk to our health. It would cause coronary heart disease, atherosclerosis, increasing the rate of heart attacks. Okay, so why do people do this in industry? Well, the thing is that it tastes good. That's one. It tastes really good. Okay, that's why many people love junk food. Or, I hate to call it junk food, but that's what they call it. It's food at the end. So it's really bad food for our health, and alhamdulillah, I've stopped eating those things, alhamdulillah. So it tastes good, and it lasts for a long time as well. It has a long shelf life, so it's good for industry. It saves money, right? Okay, so here's an example of something used in industry. So the problem with butter is that it is solid, right? So when you spread it on a piece of bread, it gets ruptured. صح بتمزعها. So it's really a pain in the neck to spread butter on a piece of bread. So the idea is that they thought, well, why don't we take something that is oily, okay, liquid form, and hydrogenate it so that we can convert it to something solid, and we can make it sort of like semi-solid so that we can spread it easily on a piece of bread. And they came up with what is known as margarine. Okay, so they came up with what is known as margarine, which looks like butter, except that the problem was that in the past it created trans fat. Okay, so but now they improved on the manufacturing of these things. So now you see on these labels that they do have 0 grams of trans fat. Okay, so pay attention to these things when you purchase such stuff. Okay, so fatty acids can be conjugated to alcohol groups or alcohol moieties, long hydrocarbon chain alcohols, okay, forming wax. Now, wax is insoluble in water. Okay, forget about this negative to acrolein test. They are not easily hydrolyzed. They are not digestible, so we can, if you eat like a candle, for example, if you're really hungry and you eat a candle, it's useless. You will not benefit from it whatsoever because you cannot digest it. They are resistant to rancidity, meaning that they do not get rotten. Okay, and they have no nutritional value because you cannot digest it. But it's found in nature. Okay, so basically honeycomb is beeswax is basically is a type of wax. Okay, the wax that we use to make candles, the wax that we use to polish cars and polish whatever furniture and whatever, it's all a wax. That's how they are made. So basically here you have a palmitic acid that can react with a hydrocarbon chain of an alcohol molecule, forming an ester bond right here, okay, linking the two to each other. Now, even though you have a hydrophilic group, still it is insoluble in water because of the long hydrocarbon chains on both sides. Now, so let's talk about membrane lipids. Now, this is basically the triacylglycerols and wax. So let's talk about membrane lipids. So there are 3 major components of membrane lipids, okay, and they are glycerophospholipids, we have sphingomyelin, and these two are phospholipids. Okay, and then we have glycolipids as well, and the glycolipids and sphingomyelin are known as sphingolipids. Note the backbone. So we have glycerophospholipids that contain a glycerol backbone in addition to two fatty acids, a phosphate, and you have whatever group that is attached to that phosphate. Now, sphingolipids, they contain a sphingosine backbone in addition to one fatty acid, except that with sphingomyelin, you have a phosphate and choline, and with the glycolipids, you have a sugar attached to the sphingosine backbone. Okay, now, so glycerophospholipids, you have a glycerol molecule, you have again one fatty acid, the second fatty acid here, and to the third carbon, which is now number 1, by the way, when you number carbons in a glycerophospholipid. So you have a phosphate that can be attached to it, okay, and then any other group. Now, if there is a phosphate only with no group, it's known as phosphatidic acid. So if I tell you what is the simplest glycerophospholipid, you say it is a phosphatidic acid. Now, to the phosphate, you can attach any other group. So you can attach an ethanol amine. Okay, so it looks like this, an ethanol amine, two carbons and an amino group attached to it. You can have a choline right here, 2 carbons, and you have a nitrogen, and linked to that nitrogen you have methyl groups. And or serine, serine is an amino acid, phosphatidyl serine. Okay. So we have different types. We have phosphatidic acids. These are the basic or simplest glycerophospholipids. We have phosphatidylcholine. These are known as lecithins, and they are the most abundant membrane lipid. Okay. Now, if you grab something like an ice cream and you look at it, you will find that it contains lecithins. So it is used in food production. We have other types like cephalins, which are both phosphatidylethanolamine and phosphatidylserine. Now, phosphatidylserine is abundant in brain tissue. We have phosphatidylinositol, so we have a sugar molecule attached to it, and it's known as inositol. Now, phosphatidylinositol is present in cell membranes, and it's important in signaling. Now, we have other types, specialized types like cardiolipins and plasmalogens. So, lecithin or phosphatidylcholine, it's the major component of membrane lipids in our cells, and it is the target of an enzyme known as lecithinase. So it breaks lecithin. And this enzyme is present in snake venom. And this is how snake venom can kill us. Because what happens is that this lecithinase would break up the lecithin or the phosphatidylcholine in the membranes, in the plasma membranes of red blood cells, hemolyzing red blood cells. Okay. Now, lecithin is really used in industry, food industry. Why? because it causes a process known as emulsification. So emulsification is basically dissolving fat in a hydrophilic environment, and it's used in the production of ice cream, chocolate, mayonnaise, and so on. So how does it work? Well, remember that these phospholipids have 2 fatty acid chains on one side and a phosphate on the other side. That's how they're drawn. So the fatty acid chains would surround the fatty droplets in these food products, right? And you have the the phosphate head is exposed to the hydrophilic environment. So what happens is that these fatty droplets are dissolved now in the hydrophilic environment. So as I said, we have specialized types of complex lipids. We have a cardiolipins. So from the name, you can tell that it's really found a lot in cardiac tissue, primarily or specifically in the inner membrane of mitochondria. Okay, so it may have functional significance. Now, it's made of 3 glycerol backbones, 4 fatty acids, and 2 phosphate groups. And we have another type known as plasmalogens. Now, plasmalogens are present in cell membranes of specialized tissues as well, or certain tissues like brain, muscle, liver, semen as well. They have protective role against reactive oxygen species. What are reactive oxygen species? Basically, these are oxygen-containing chemicals that are highly reactive, meaning that they like to participate in reduction oxidation reactions, so they like to be reduced, oxidizing other molecules, and these molecules can be damaged. Now, an example is hydrogen peroxide, H2O2. Another example is oxygen radicals, so molecules known as oxygen radicals. These molecules are missing an electron, so they want to oxidize other molecules. They want to steal this electron from these other molecules, including lipids and fatty acids, okay, damaging these molecules. Okay, so what happens is that plasmalogens is one of those molecules that would sacrifice itself for the sake of protecting the cell and other molecules. Now, what's important about plasmalogens is that its backbone, or the precursor, is dihydroxyacetone phosphate. It's not glyceraldehyde 3-phosphate. So the backbone is not a glycerol or derived from glyceraldehyde, rather it is derived from dihydroxyacetone, the other isomer. So don't you love the sound of music? Amazing. So, so that's the precursor. Now, it contains a fatty acid chain at carbon number 1, and it's connected to an ether group. Okay, so there's an ether group right here. And at carbon number 3, there is a phosphate, and you can have either ethanol amine or choline attached to it. Now, there are different types of plasmalogens. So you have ethanol amine plasmalogen, which is present in the nervous tissue. You have choline plasmalogens, which is mainly present in cardiac tissue. It's also known as, it is also a platelet activating factor during platelet aggregation. And you can have serine plasmalogens as well. So there are different types of plasmalogens. Now, note the ether group right here, and the alkene as well. Okay. Now, we have another phospholipid known as phosphatidylinositol. Note that you have a sugar group attached to the phosphate, and phosphatidylinositol is basically a molecule that's present on the cytosolic side of cells, and it participates in signaling. Okay, so how does it participate in signaling? Basically, it can be hydrolyzed by an enzyme known as phospholipase C, producing 2 molecules known as diacylglycerol and inositol triphosphate, and they have important signaling, they activate signaling pathways. We'll talk about this later on, maybe in this course or in the endocrine course. So there are different types of phospholipids. Okay, so phospholipids can form micelles. Remember these micelles? Soap, same idea. So you have the phosphate group to the outside, exposed to the outside, the fatty acid chains hidden inside the ball. You can have what is known as a liposome. Now, in this case, you have double layer of phospholipids. So you have these phospholipids on the outside, and you have them in the inside as well, interacting with each other. And you have two layers of fatty acid chains. Okay. Now, if we extend this, by the way, it would look like a bilayer sheet, which is the basic structure of membranes. In fact, if you look at a whole cell, it looks more like it's a liposome where all organelles are inside. Okay, but the cytosolic part that is the inside, the internal of this liposome, is larger than what you have in here. Now, what's important about liposomes, by the way, is that they can be used in delivery of molecules, other molecules that are hydrophilic or ionic. So the idea is that we can enclose these molecules inside a liposome. And when we add liposome to cells, this liposome can integrate itself. It fuses with the plasma membrane. It becomes part of it, releasing these contents inside cells. So this is one way by which we can actually in labs deliver DNA and RNA inside cells. And it's one of those mechanisms that one of those mechanisms that can be used in pharmaceuticals as well in delivering drugs to cells. So let's talk about sphingolipids now. So sphingolipids, they have a sphingosine backbone. This is what a sphingosine looks like. Note that you have an alkene group right here. Okay, so try to differentiate between plasmalogens and sphingosine in structure. So in a sphingosine you have an alkene, but it does not have an ether group like plasmalogens. And there is a fatty acid chain that is attached to an amide, or via an amide bond right here. So this is an amide bond. Okay, and then you have at carbon number 1 you have any group attached to it right here. Okay, now, so if I bring you a structure of a sphingolipid and I ask you what is this molecule, you need to differentiate it from glycerophospholipids and other molecules by looking at certain features. Again, you have a double bond in here, an alkene. You do not have an ether group like plasmalogens. You have an amide bond, and you have a fatty acid chain attached to it, and then you have another group attached to carbon number 1. Okay, so why were they called sphingolipids? Because when they were first discovered, they thought that these were mysterious molecules like sphinx, Abul Hol, and Ahram, Abul Hol. So it looks mysterious like sphinx, so they called it sphingolipids. Okay, funny, huh? So the basic sphingolipid is known as a ceramide. Okay, so right here you have a sphingosine, that's the sphingosine backbone right here, and to carbon number 2 you have attachment of a fatty acid chain. Okay, right here, this one in blue, and that is known as ceramide, the basic structure of a sphingolipid, or the simplest. Now, there are different sphingolipids, as I said, depending on the group that is attached to carbon number 1, either a sphingomyelin or a glycosphingolipids, or known as glycolipids. So sphingomyelin has a phosphocholine. Okay, so that's carbon number 1 right here. This is the sphingosine backbone. Here's the alkene right here. So we flipped it, okay? So pay attention to that. It's flipped. Now here you have the amide bond right here, linking the fatty acid chain to the sphingosine, and then you have a phosphocholine. This is phosphocholine right here. Okay, so this is sphingomyelin, which is basically it creates, or it forms, the myelin sheath that is present in the central nervous system, protecting our neurons. Okay, so that's how it looks like. Okay, so here we have a neuron, and right here you can see the axons. You can see the myelin sheath covering it. Right? Zoom in, and you will find layers of myelin sheath right here, and the axon is right in the middle, protected. Zoom in further, and you will find that you have layers of the bilayer sheath, and inside these layers there are these sphingomyelins right here, and you have myelin binding proteins and whatever. So you look at one of these, that's how it looks like. Okay, so that's a the sphingomyelin right here. So again, look at the structure. This is the sphingosine backbone right here. These are this is the alkene molecule right here, and you have the phosphate group, and attached to it you have the choline group right here. Okay, so how about glycolipids? Glycolipids have a they have a sugar group attached to the sphingosine backbone, and there are 3 types of glycolipids depending on the nature of the sugar that is attached. Now, so these glycolipids are important because they differentiate cells from each other. They can participate in signaling and etc. Cell recognition and so on. So there are 3 types of glycolipids, and these are known as cerebrosides, globocytes, and gangliosides. From the name, you can realize that they are rich in the nervous system: ganglions, cerebral, and cerebellar, and so on. Cerebral, whatever. Anyhow, so cerebrosides, they are the simplest glycolipids. They contain a single hexose. It can be a galactose or it can be a glucose. Okay, so right here you have a glucose attached to the sphingosine moiety. Look at carbon number 4. It's going down. That's how we can differentiate glucose from galactose, and this is galactose. Okay, so right here with galactose, carbon number 4 is going upward, so that's galactose. Okay, so globosides is basically when you have more complex lipids. Okay, like this one right here, for example. Okay, so you have a number of sugar residues attached to the sphingosine. You have a chain of lipids. Now, the difference between globosides and gangliosides is that gangliosides contain sialic acid, and remember that this sialic acid would be the very last sugar attached to that chain. Now, gangliosides are targeted, by the way, by the cholera toxin in human intestines, and that's why you have the effect of cholera toxin. Now, you can have galactocerebrosides or the sugar molecule, the hexose, can be modified by the addition of a sulfate group as found here, and this is also a component of the myelin sheath, and it's negatively charged and so on. Okay. Remember how we talked about blood groups and we said that we have type A, type B, type O, and so on? Well, the thing is that's based on glycoproteins, right? We talked about that. But you can also have glycolipids that participate in this typing, blood typing, okay? So it's not only the sugars that are attached to proteins, but also the sugars that are attached to the sphingosine. Okay, same chain of sugar residues attached to the sphingosine as in glycolipids.